our first speaker, who is John Hartwig, the Henry Rapoport Professor of Chemistry at Berkeley. John had previous appointments at the University of Illinois, Urbana-Champaign, and at Yale. He received his PhD in 1990 from Berkeley. He's known for his work in organometallic chemistry and organic chemistry and catalysis, largely applied to developing new methods in synthetic organic chemistry. He has many recognitions of his the excellent caliber of his work over several decades, including election to the National Academy of Sciences, and the only other one that I'm going to mention is that he was a 1997 Camille Dreyfus Teacher Scholar. John.
Okay, well, thank you, Matt, for the introduction. Thanks to everybody who was a part of organizing this event. Congratulations to many of you who have received Teacher Scholar Awards. I just wanted to point out a few connections. Myself and the Dreyfus Foundation, so I received a new faculty award in 1992, which helped my group get started. And then the Teacher Scholar Award that I received in 97 really created an inflection point for the development of one of the projects in my group that I'm going to talk about here in just a few slides, but was one of the projects that I think we're most known for on the cross-coupling to form carbon-nitrogen bonds. So we were asked to give a relatively general introduction, maybe overall a very general talk to palatable, at least to a non-chemistry audience. I know there are some people here who are not chemists. I know there's an atmospheric chemist somewhere in here that I met at breakfast, so there are people who think about chemistry that's very outside of what I do. Sometimes people say, oh, you should be able to describe what you do to your parents when you go home for vacation. Well, this is reality because my mother's in the back of the audience here, so I have to do quite a good job here today of creating a talk that can be understandable to someone who is a music major in college. So I don't know if I'll succeed at that, but I hope that I can tell you some things about how what we do in my lab, but maybe more so what synthetic chemists do in general, what some of the challenges are for that field, and how what we do is trying to change the way people make molecules, think about making molecules, but maybe even more so, change the molecules that people decide to make because of the development of new synthetic methodology. So that's what I wanted to talk about today. So I know, again, most people here are chemists, but only a few are synthetic chemists. So I thought I would start with a slide just showing how many things around us are made from chemistry, and essentially everything is. And in a couple of slides, I'm going to reconnect to that video as to why we're all here today, actually, what reaction led us to be here today. So anyway, whether it's medicines or medical devices, whether it's feeding ourselves, we make about 500 billion pounds per year of ammonia that becomes fertilizer. And if you think about your own bodies, that means that half of the nitrogen atoms in your body are made through chemical synthesis. And that chemical synthesis, as a theme in the talk today, is going to be through catalysis. So one of the classic catalytic reactions is the reaction of hydrogen plus nitrogen to make ammonia that ends up in your body from plants to animals to you. Of course, there are polymers. You'll hear about some high-tech polymers today. Some of them are for safety and for helping us feel good, for lightweighting vehicles, for allowing us not so much polymers, but materials to give us alternative energy sources, to help us communicate. And even things that seem like they're from a natural source, like a piece of wood furniture, actually has coatings that are often made through chemical synthesis. So I thought I'd take one example of where chemicals come from to sort of introduce how catalysis can really change the way molecules are made. So let's take acetic acid, right, which is vinegar. So vinegar is a natural product, after all, right? It's made through fermentation. You get it from sugar. But we can't make all of our acetic acid from sugar because the chemical industry requires about 5 billion pounds of acetic acid per year. So that's a lot of sugar, which is a lot of food that we don't want to use to make that. So instead, it's made from coal. So how do we get from coal to acetic acid? Well, coal treated very high temperature with water makes so-called syngas, CO and hydrogen. And then in one of the largest scale catalytic processes, that's reduced, hydrogenated to make methanol, which is wood alcohol, but is here made through catalysis. And then a homogeneous catalytic process, a rhodium catalyzed and now iridium catalyzed reaction makes acetic acid. And so if you go to the stockroom and buy acetic acid, that's how it's made through this process. If you go to the grocery store and buy it, it's made this way. Okay, but you heard a lot about cellulose acetate in that video. And so how was it originally made? So it turns out the Dreyfus brothers made that from butane. Right, so a really remarkable catalytic process, the oxidative cleavage of the carbon-carbon bond, making a C2 product that's acetic acid. And so I was recently a Dreyfus lecturer in Basel, which was a great honor, like the honor it is today to speak in front of this audience. And became familiar with the really origins of the chemistry that they developed to be able to make the cellulose acetate and grow this company called Herc Celanese, which was actually the first industrial trip I took to give a seminar. I was at Herc Celanese, or Celanese originally, but then became Herc Celanese. All right, so that's a natural product, but when we think often of natural products, we think of very complicated molecules, one famous one being vancomycin, that is a sort of a last resort antibiotic that has been made, it's a natural product made by nature, it has been synthesized, but in 36 steps by Dave Evans, among other people. Taxol is another one, so that's one of the most prescribed anti-tumor medications, chemotherapy treatments. And so that was originally made by the Pacific yew tree. Those are precious plants, and so now it's made by the European yew tree and by chemically putting on the side chain. But originally, chemists like Paul Wender at Stanford made that molecule, but by 40 chemical steps. So that's a lot of reactions, it's a lot of time, and a lot of energy that goes into human energy that goes into making those kinds of molecules this way. So could we do it in a better way? But maybe more important is not so much making molecules that nature already makes, but making molecules that nature does not make, right? So let's take the statins as an example. The statins were originally natural products discovered by two different people at two different companies. And those were medications for a while, but actually, the best-selling drug of all time is Lipitor. Lipitor is not made by nature, right? Chemists designed and synthesized and still make that molecule, sort of inspired by these, but this is the structure of Lipitor. And so we need to be able to make molecules like Lipitor on large scale. So let me show you how Lipitor is made. Probably you can't read this. It's supposed to be very small. Just to point out that there's many, many steps. In fact, there's 16 chemical reactions, 11 isolations in between that for at least original synthesis of process synthesis of Lipitor. And I won't ask you to look at all the reagents and all of these on these steps, but I just want to point out that almost every one of these steps could be understood by an undergraduate when they graduate with a chemistry major. They're not particularly sophisticated steps. Maybe one of these, the Stetter reaction here through a carbene intermediate, would be something that's less familiar, but mostly these are very standard organic reactions. So can we make these kinds of molecules more efficiently? And so that's where catalysis comes in, the field that I work in. So maybe you know your body does chemical synthesis with enzymes that are catalysts. And I showed you about the acetic acid synthesis. A plant that would make acetic acid is not exactly this one, but would look something like that, very large scale, making billions of pounds of material. But those statins, or here I show Indinavir, Crixivan, the first drug that was actually had any effectiveness against HIV/AIDS, is a pretty complicated molecule with many stereocenters, developed at Merck and then synthesized in very large scale by the Merck process group. And so this involved many catalytic reactions, including enantioselective reactions. And so a current goal is to make these kinds of molecules, right, with the kind of catalysis that is done in your body or with the efficiency that's done for these very large scale processes. So my last sort of introductory note is on how catalysis can have a strong influence on human health. So we can maybe make Lipitor in a better way, but actually catalysis changes the molecules people make. And one example would be this drug called lorcatin. So it's an antihypertensive drug. The structure is not that complicated, and actually maybe to some people in the room it looks pretty easy to synthesize because we can make that carbon-carbon bond very easily. Okay, that's a catalytic reaction. It's now a famous catalytic reaction called cross-coupling that was the subject of a relatively recent Nobel Prize. And so because that carbon-carbon bond can be made, this synthesis is now easy, right? You make that core of that molecule, and now it's just, again, sort of sophomore or maybe junior level in organic chemistry to make the rest of the molecule. But imagine whether making these sartan kinds of drugs if you couldn't make that bond. So you could buy biphenyl, and then you'd have to put one methyl group on the top and one here. I don't know any way to do that. Nobody can do that. Right? And then from there, you would do the rest of the molecule, but you would just never choose to make those molecules. So there's a whole array of billion-dollar drugs, these sartan drugs, that are on the market and were made by discovery chemists because they could make that carbon-carbon bond. All right, so let's talk a little bit more about catalysis before I go into some specific results from my group. All right, so here's the periodic table, familiar to all of you. These atoms, these elements on the top right, those are sort of the elements of organic chemistry. But we're going to put those elements together to make molecules that have function, whether it's for medicine or some of the materials you'll hear about in later talks, using metals like these, rhodium, iridium, platinum, ruthenium, relatively expensive precious metals, but incredibly valuable in the chemical sense for doing organic synthesis. Now, we're trying to do things these days with cheaper first-row metals, but if you go all the way to the left side, these are not often used to make the molecules I'll talk about today, but are used to make polyethylene on a scale, again, of 500 billion pounds per year through titanium and zirconium catalyzed chemistry. Anyway, how does a catalyst work, right? We have a starting material, we have products, we have an energetic barrier to get from the starting material to the product. What does a catalyst do? So it lowers the barrier, reduces the energy, so it makes the rate faster. And of course, a catalyst is not changed by the reaction, so it doesn't change the energies of starting material and product because it's not changed by the process. Okay, but what a catalyst actually does is not simply lowering that barrier, but to create a completely new pathway by which the reaction occurs, right? And it is often many steps, but all of the barriers for those steps are much lower than the highest barrier. All right, so what are the challenges in this field? What are the overall goals of this field? And how does what we do fit with those goals? All right, so I'd say general goal number one, there are many, I'm just going to talk about two, is to change the strategy and targets for chemical synthesis by discovering new reactions, like that carbon-carbon bond forming cross-coupling reaction that leads medicinal chemists to make biaryls so frequently in their drug discovery efforts. The second goal is to take something that's often a very empirical field and turn it into something that's more of a design process, something that's built off fundamental principles. All right, so this is what we do. This is the kinds of catalysts that we work with. All right, so if in a general sense, you'd have a metal in the middle, you'd have some reactive ligands on it, and then let's call them the controlling ligands, these other ligands on the outside. Here's a space-filling model of one of those. The peach in the center is palladium. Right? And all of this black and white material on the outside are the carbons and the hydrogens that are the controlling ligands. Here's an aryl group, and here's a halogen, and the peach in the middle is the palladium. So that's where the reactivity occurs, the so-called active site. But all of this around the outside is the key to controlling how this chemistry occurs. And so how do we decide what to put on the outside? Well, what my group does is to try to follow an approach where we use information from understanding reaction mechanism by physical organic chemistry kinds of methods to understand how the structure of this affects reactivity, what the individual steps are of these catalytic processes. We also are interested in new experimental designs that help us to discover catalysts faster. And finally, we're also just interested in discovering completely new types of catalysts, like I'll talk about at the very end of this lecture. All right, so let me go back to the beginning of my group, the Dreyfus teacher-scholar era of my lab. All right, so one of the reactions that we were interested in studying is the one shown here that was originally published by two Japanese chemists, Kosugi and Megita. And it was a reaction that would make the carbon-nitrogen bond in an aromatic amine by taking an aromatic halide and a tin amide. Not a very practical reagent, but to us seemed like an interesting reaction from a mechanistic standpoint, but also for synthesis. All right, so why would this be important synthetically? Now, as a quick interjection, I don't want to digress too much for time reasons, but I was getting a lot of flack for why we'd want to do this, because you can simply take a nitroarene and make aromatic amines from that. Okay, and I'll get back to that in a second. But the Dreyfus Foundation funded us for this work and allowed us to develop one of the 20 most used reactions by medicinal chemists today. All right, so why is this important? Well, maybe you remember from sophomore organic chemistry, you can do nucleophilic substitution on an alkyl halide. But that you can't do this on an aromatic halide unless it's a special one. So this reaction needs a catalyst to make it work. All right, so why would you want a catalyst for that? Because, as I said, you can make these molecules just like you can make biaryls by some means or another, just not quickly. Right? So you can make aromatic amines. Right? How do you make them? Well, you just take benzene, pulling out of an introductory organic course, nitrate that, reduce it. Maybe you put a blocking group and then alkylate, take the blocking group off to get good selectivity. Okay? But from here to here, that's 1, 2, 3, 4, 5 steps. Maybe 4 in a different way, but 4 or 5 steps. So catalysis can make this more efficient. You can do a halogenation of an arene, right, and then take that aryl halide and do a catalytic substitution. But maybe more important is that this is a much more versatile way to do this than on the top, right? Because there are so many aromatic halides. Now you can buy 200,000 aryl chlorides commercially available from various sources. And so this is actually a one-step synthesis from a very wide variety of materials. Whereas nitroarenes, if you want to start from that, there's going to be a few thousand of those, not hundreds of thousands available. So this reaction then becomes important for maybe all of these sorts of applications if we could do this in a way that's much more practical and overcomes all these original problems. Our interest was largely mechanistic in the beginning, and so I'll touch on that, but it's turned out to be very important synthetically. And so my group and Steve Buchwald's group at MIT began to study this chemistry around the same time, and we both published on this now much more practical way to do the reaction, which is an aryl bromide and an amine with a base and a palladium catalyst to make the carbon-nitrogen bond. One step with all commercially available materials. All right, so I won't go into the details of this chemistry. I want to talk about some other projects in more depth, and I won't have enough time to go into all of Steve's contributions. But just to point out a couple things from my lab that show how this could become a very efficient and very synthetically useful reaction. So the first was to show that we could take a catalyst that had very large groups on the phosphine, a single phosphine per palladium, and be able to do a reaction, this amination reaction, on an aryl chloride at room temperature with just a couple minutes worth of reaction time. Okay, so the hindered phosphine made very highly active catalysts with 1 phosphine per palladium. But the scope was limited at that time to secondary amines, so 2 R groups, 2 alkyl groups, or an aryl and an alkyl group on the nitrogen. So my group and others were seeking to be able to increase the scope of this reaction to be very selective for primary amines. So to do this, we tapped into some mechanistic information that we had gained from my group. And so we wanted to understand how the carbon-nitrogen bond would form. And so we had made a series of palladium complexes. This is one of our, I guess, best known mechanistic studies is to make aryl palladium complexes with amido ligands, diarylamides, arylamines, alkylamides, and show that for the bottom example, the one down here, that we could form the carbon-nitrogen bond from an amide derived from a primary amine with not perfect yield, but pretty good yield of that product. And we did so by having the 2 phosphines linked together in these so-called chelating ligands, and that controlled the side reaction of beta hydrogen elimination we were trying to prevent. And so by changing some of the structural features of that, putting more hindered groups on it, putting more donating alkyl substituents at phosphorus, changing the architecture of the backbone a little bit, we then were able to take aryl chlorides with primary amines and make the arylamines at room temperature. And the catalysts were very stable, so we could actually get about 20,000 turnovers of product. So you take like an aspirin-sized tablet and make a big bag full of product, and that is efficient enough for it to be used on process, right? To make things like pharmaceuticals. And so this compound is a antidepressant that was recently approved by the FDA, and it was made, maybe not in its final way, I'm not sure, but it was certainly in discovery routes and to bring molecules into clinical trials through to carbon-nitrogen bond formation, carbon-sulfur bond formation, 2 palladium catalyzed cross-coupling reactions. Right? And so this molecule becomes very accessible to at least the discovery chemists by having these types of coupling reactions available. All right, so that's a snapshot of Project 1, but that led us and others in the field of catalysis to wonder what are we doing with all these functional groups? Why are we using so many functional groups in this chemistry? Right? Do we really need all of them? And so if you look at a sophomore organic textbook and look at the chapters of that book, many of these chapters are sort of a march through the various functional groups of organic chemistry and how we can interconvert them. Not so much how we put them into a molecule, but how we interconvert them and take one functional group and turn it into another functional group, like taking an aryl halide and making an arylamine from that. So we've wondered, along with others in the field, as to whether we could kind of create a new way of thinking about organic chemistry that would be less reliant on the functional groups that are all of these chapters of this book. Not that we need those functional groups in the end for the function of the molecule, but maybe we could do a synthesis that directly installs them, and installs them then at the position of a CH bond, which is the architecture or the skeleton of a molecule, not the functional groups. And if you think about how this is done in nature, right, there are P450s in your body, right, that will do hydroxylations and oxidations of CH bonds in the biosynthesis of some of important molecules that are natural products. But as chemists, we don't simply want to make carbon-oxygen bonds. We want to make CN bonds, CC bonds. We want to be able to put these kinds of functional groups into molecules like this. So that's in part where the field of CH bond functionalization has arisen. So could we take molecules, maybe not quite as complex as this, but be able to take the position of a CH bond and install functional groups directly? Now, in parallel with that cross-coupling chemistry I just talked about, my group was working in this area and had a sort of serendipitous finding that if you photolyze these tungsten compounds that had carbon or metal boron bonds, that the carbon monoxide would dissociate, and a CH bond at the end of an alkane would be cleaved. And this ligand that we'll call BCat star, catecholate on it, gets transferred to the terminal position of the alkane. And that's the strongest CH bond in the molecule. And to this day, there's no other reagent in chemistry that will functionalize the strong primary CH bond of an alkane in preference for the secondary CH bond. So we then, from that, developed a catalytic process, right? And the catalytic process then involved this commercially available reagent, which is the source of the boryl groups that goes onto the metal center, with an alkane, and now with a rhodium catalyst that would react with this, then cleave the CH bond, then extrude these products and go back and start over again and do it over and over. And so this is a catalyst now that will functionalize the strong primary CH bond at the end of an alkane. Right? And again, I think there's no other catalyst that will do that. Now, I showed you in the intro slides, right, the Dreyfus brothers cleaved a CH bond and made acetic acid, right? But that was the secondary CH bond. Those guys couldn't cleave the primary CH bond, but we can. So anyway, we could go from alkanes to these alkyl boronate esters. All right, so why do you want to do that? Why would you ever want to put boron into a molecule? Because after all, that's boron, and boron is ant poison, right? So boric acid is ant poison. Why do we want to put ant poison at the end of a molecule, right, or the equivalent of that? And that's because, again, there's sophomore organic chemistry at carbon-boron bonds by which you can oxidize that and make an alcohol, or actually do now a whole array, probably 25 or 30 different reactions that can be done at carbon-boron bonds. The only problem is that this is not very practical as I've drawn it, because we're making an alcohol, a linear alcohol like butanol or octanol, which is higher value than the alkane, but still much lower value than this diboron reagent. So economically, this makes no sense whatsoever to do. So how could we make this practical? And so where this became more practical is the functionalization of aromatic CH bonds. And the importance of this reaction is the selectivity. The selectivity for the most sterically accessible CH bond, which after all, the primary CH bond's mostly the most sterically accessible, which is why the reaction occurs there. In this case, it's also the most sterically accessible CH bond that reacts. But now we're going to take things that have much higher value and do chemistry on those. And so here's just an array of examples from the reaction of an arene with the diboron compound to make the aryl boronate esters, as they're called, and showing that the reaction occurs in all of these cases at the sterically most accessible CH bond. And these molecules have a number of functional groups already on them, right? There's a nitrile, and somewhere in here, so there's an aromatic nitrile, and there's halides. All of the halides are on here. There's an ester. But the reaction doesn't occur at the functional groups like it does in sophomore organic chemistry. It occurs at the CH bond instead, replacing the boryl group at that position. And we can do this very efficiently. So what could we do with these? We could do those 25, 30 different reactions to make different products for this, including a phenol product. More recently, we've done this with silanes and work that was just recently published. And to some extent, that's important because you can buy the silane that we use for a couple of dollars a kilo. So that starts to make this chemistry look even more economically attractive. And the products that we make from functionalizing heterocycles are more stable than if we were to install a Boryl group at those positions. So I just borrow one example of an application not from my group, but from the Merck process group. And so this was published recently in an ACS journal, Organic Process Research Development, in which they make they run the chemistry on a 100 kilogram scale. And this first step is that iridium catalyzed borylation. The second step here is the oxidation. And so they take this arene, just like the ones I showed 2 slides ago, and put a hydroxyl group in the position that you would be real challenge to install the hydroxyl group by sophomore organic chemistry, because these are orthopara directors on here. And so that's a reaction that goes at the position where you wouldn't get the installation of the group from electrophilic aromatic substitution. All right, so I've shown you now a couple of versions of catalytic chemistry, one with palladium, one with iridium, reactions that have nothing to do with biosynthetic pathways, but making molecules, not so much in my lab, but applications in other labs that have a number of functions, whether it's for making whole transport layers of organic light-emitting diodes, or making medicinally active compounds or agrochemically active compounds. And by just as a few examples of how we show we could directly install functional groups in those molecules in the second part. So we can put the Boryl group at the end, and we can put, I didn't show you, but silyl groups along this chain at different positions. But could we install a functional group anywhere we want in a molecule, not controlled by where the hydroxyl group is, or to change the selectivity from what you'd get from an iridium or rhodium or palladium small molecule catalyst? So we've recently been asking the question of not just could we install a functional group at a position where it doesn't exist, but could we select where that functional group goes? So let's go back to these P450s. That's what P450s do, right? They install functional groups at various positions that are needed for to make molecules that have biological function. Now, maybe some of you know that this year's Nobel Prize was given for directed evolution, and that to Francis Arnold, among others. And in Francis Arnold's group, they've worked on some of these P450s by changing the substrates for those enzymes and the position at which the reaction occurs. So could we take that sort of capability together with organometallic chemistry to be able to direct a reaction to different positions on the molecule? So how would we do that, right? So that we could take these groups instead of hydroxyl groups and put them in molecules. And so that's one project that we've been working on in my group most recently. And so how would we do this? So the idea is that we would take some heme protein, like a P450, and we'll start with myoglobin, and change the metal that's inside those proteins. Okay? And so we're going to change it from iron that does its own chemistry to something like iridium that does different chemistry, or maybe faster than iron does. And so we're going to change that metal, and by changing that metal, we can change the reactivity, and maybe that reactivity could be modulated by a side chain from an amino acid in this so-called axial ligand that's bound to, that controls whether a protein carries oxygen or does catalytic oxidations. And then we're going to potentially use directed evolution to modify the shape of this binding site so that we could select for a small molecule over a big one, or a big one over a small one, or have it react at one position or another position of that molecule. So that's the plan, but you can't simply go into a heme protein and extract the iron atom out of the middle of the porphyrin that's in the middle of that protein. So we really need to kind of build this up rather than just start with a heme protein. And so the strategy we started with was to express the protein under conditions where we would have low iron concentrations, and also the broth would be missing the aminolevulinic acid, the precursor to make the porphyrins. And so then from that we would make an apoprotein. Okay, an apoprotein is a protein that's missing its metal, or missing, in this case, the whole cofactor, no porphyrin, no iron. That's why it's clear. And then we can add the iron back in, or we can now add synthetic metal porphyrin complexes that would contain rhodium or copper or manganese, cobalt, or iridium or rhodium. And so that's what we did. And with the reaction we sought to catalyze was a CH activation reaction that would have nothing to do with biology or nature or biosynthetic pathways. That would be a carbene precursor. This would lose dinitrogen, make a carbene precursor that would insert into the CH bond. Okay? And so that was our target reaction, because it was kind of like inserting an oxygen atom into a CH bond or a nitrene into a CH bond that we knew some enzymes could do. And so that's the reaction. These are some of our sort of pictorially described results. So here we've made mutations to the called axial ligand, or the position of that axial ligand, to things that could be ligands, or in case of alanine or glycine, just a small group or no group that's going to leave space in the protein for a ligand that's on the metal itself. And so the results here are that the darker the color, the higher the turnover numbers. And the winner here, you can see, is iridium, but not just iridium with a methyl group attached to the iridium. So we've actually inserted an organometallic site with a metal-carbon bond into a protein to catalyze this CH activation, functionalization. So that's our cofactor that's in the protein. So that's the good news. The bad news, actually, and sort of nail-biting time period, was that the product of that reaction was racemic, right? Equal amounts of the right and left hand of the molecule to enantiomers. And so proteins are chiral. And if this reaction's occurring inside the protein and it's a chiral catalyst, you should be making one enantiomer over the other preferentially. But it turns out that I think we just didn't have the right shape to the binding site for the organic molecule, because after just a few rounds of evolution, what we could do is to favor the formation of one enantiomer versus another enantiomer in this kind of evolutionary tree I show here, where we make a mutation to position 93 to glycine or alanine, and then make mutations to the position above the metal active site, and then successively in the active site further and further away from the metal, and evolve the system to be able to get, not perfect, but reasonable enantiomer selectivity for one enantiomer, and also evolve to make preferentially the other enantiomer, although not with that high a selectivity. So that showed that we could follow this strategy to make an artificial metalloenzyme that would catalyze an enantioselective reaction and evolve it to improve that EE. But I began my talk, or a section of my talk, talking about P450s, which are enzymes. Myoglobin's not an enzyme, right? Myoglobin binds oxygen in muscle, and it has no site for an organic molecule. So it was kind of surprising that we could actually, well, reasonably surprising that we could get this to give good enantioselectivity for something that has no binding site for an organic molecule. So our second generation was to use not myoglobin that I show here, but a true P450, a real enzyme, right, that has a binding site for an organic substrate within it. And that's a P450 called CYP119. That's from a thermophilic organism, so in principle we could do reactions that would occur at higher temperatures. So let me just give you a snapshot of some of our latest results on this. So first, we have a lot of improved enantioselectivities, high turnover numbers, good stability out of the P450 we generated in the same way as we generated those myoglobin systems. But for the point I want to make about being able to make a reaction occur any position that you want on a molecule, I point out that one of those mutants of the P450 based on CYP119 could catalyze the intermolecular insertion of a carbene into a CH bond. Now, admittedly, a little special CH bond being benzylic and next to oxygen, but still an intermolecular reaction to make that product. Now that molecule is symmetric, so every CH bond at this methylene and that methylene is identical. But in this compound, and others like it, where we put a substituent off the back, one substituent makes this methylene group different than that methylene group. And I think it would be hard to have a small molecule that would select one over the other with just some substituent off the back. But maybe with an enzyme pocket, you could orient that molecule in a certain way to control that site selectivity for this site versus that site. And by directed evolution, you can improve that selectivity and maybe reverse that selectivity. And so although our numbers aren't perfect yet, what we show here is that we can select with one of the mutants, the CH bond that's para to the fluorine, and with another mutant, select for the CH bond that's meta to the fluorine. And we're looking at doing that for other substrates as well. But I like this one in particular because you can't get anything smaller than fluorine except for hydrogen. And if you know your Hammett parameters, there's nothing that is less perturbing of electronics on an arene ring than a fluorine group. So that's the smallest perturbation we could make, and we can still reverse the selectivity by 20 to 1. All right, but if we really want to mimic nature, maybe we want to do that reaction in a cell, or reactions like that in a cell. So is it feasible, first, to express the proteins and isolate them from the cell with iridium in it, and maybe even do the reactions that way as well? All right, so the idea then is to have something that would, known transporter that would transport a heme across the cell membrane, but could take the iridium porphyrin across the cell membrane, bring it into our overexpressed mutant protein and have it bind to that protein, and either isolate it for reasons of screening and improving selectivities and changing reactivity, or maybe doing the chemistry itself in a cell. And sort of fortuitously, by having this methyl group as a ligand on iridium, right, that makes a clash with the thiolate or histidine that's on a natural heme protein, the groups that bind to iron. So the iron will bind to the other P450s that might be expressed, but the iridium is going to bind to our mutant one that has an alanine or a glycine at that position. And apparently, the bit of hydrophobicity or grease of that methyl group makes it bind selectively to our mutant protein over the natural P450s. So we actually don't have to worry about low iron conditions and all of the things that we were doing in the beginning. We can just simply grow up cells, add some iridium porphyrin to them. They take it up through that through the transporter, and we get a protein out. This is our pellet of this protein that has the color of the iridium porphyrin. And here spectroscopically, you can see there's a signal for the iridium porphyrin when we grow it under that. And if we don't put the iridium in there, there's no signal at this position for the porphyrin, so our isolated protein doesn't even contain iron in it either. So with that sort of orthogonality that's kind of reminiscent of Stuart Schreiber's bump hole ability to make things bind selectively, right, we can now think about actually doing a reaction in a cell. And so we're working with Andrila Mukhopadhyay and Jay Kiesling and at J-Bay and in chemical engineering at Berkeley to engineer cells that have a biosynthetic pathway in them. They're world-famous synthetic biologists who can then think about dovetailing with our catalysis to take the proper plasmid, put it into the cell, and then in a whole cell, do a CH bond activation and insertion of the carbene into the CH bond and enantioselectively within a whole cell by assembling this all in there and doing the reaction there. So ultimately, and we can do that with high turnovers and enantioselectivity. And so we're still a ways away from being able to do this final goal of being able to do a reaction at intermolecularly at any position on a molecule. But I hope I showed you a step forward of taking some of the small molecule organometallic chemistry that's been developed over the years and be able to apply that to putting that inside a protein and those proteins then to maybe creating artificial biosynthetic pathways that could make molecules like this that would be unnatural products, maybe someday produced within a cell. So that's what I wanted to tell you about today. People like Zhenin Liu, Hana Ki, and Pavel Didyo were the ones who developed the last project that I talked about. There have been many people who developed the chemistry I talked about on the CH activation chemistry in the middle. And the very original cross-coupling chemistry, again, was the work of many, many people. But I guess I should point out Caleb Carmel was the one who did the most recent development of silation chemistry in my group, and Chen Cheng is the one who developed the chemistry I showed on that slide. I just wanted to thank you for your attention, and it's a real honor to be able to be chosen to speak at this symposium, and congratulations again to many of you.
We have some time for questions.
This way.
Okay.
Hi.
Which way?
Great talk, John.
Oh,
I was wondering, do you have to worry about the stability of the Apo-Protein?
Yes, in fact, that's one of the main motivations of being able to directly incorporate the iridium, because maybe we just lucked out that CYP119, or maybe because it's from a thermophile and so it's sort of inherently more stable, that it could be isolated in its apo form. But many of them can be isolated in their apo form, but at ridiculously low levels of purified protein at the end. So maybe the bigger the binding site is, the less stable it is. I don't know if there's a direct correlation there, but we're trying to sort of apply this to a wide range of P450s, and so we think we have some breakthroughs on being able to directly isolate them containing an artificial cofactor that would then make them more stable.
Hi. In your early screen of the myoglobin-derived catalysts,
Yeah.
You had turnover numbers for a bunch of different metals. So many of them were working, but then they were failing. Do you know how they were failing?
We don't really. I mean, we were looking to get some starting point. We've gone back to look at, with other mutants, besides the one we started with when we did our initial screen, and also with the P450s, putting other metals back in. Iridium methyl is always the winner. The reactions are often very fast. I mean, the breakdown of a diazoester is catalyzed by many different metal complexes, and it's not like trying to take an oxygen atom and insert it into a CH bond like a real P450 does. So there are many complexes that will catalyze something, but in many cases, you get the carbene and it decomposes, but in some way. The most common way, I don't know if it's actually happening here, but the most common way is it would effectively dimerize to make an alkene. And so if you're trying to do intermolecular reactions, that's a competing pathway. There may just be some hydrolysis or, yeah. But in this game, it seems to me, protein precipitates out. You get a little bit of reaction, protein precipitates out, and then trying to figure out what happened is actually a major task, so we just moved on.
So I was wondering about, how easy it is to detect if you're making these carbenes that do insertion reactions. You might be modifying the pocket
Absolutely.
itself.
Yep.
And in some sense, it sounds like another opportunity for, if let's say if you are still getting turnover, you can prime your catalyst with something that creates even a different environment, and then with one reagent and then try another one. Have you done these sequential things with different reagents?
Oh, that's interesting,
to see
yeah.
if you can influence because it's going to be really hard to detect if that chemistry is going on, but you might be able to indirectly detect it.
Right, so we've looked for that, and so it's a short talk, so I didn't have time to go into all the background. But Francis Arnold has looked at carbene transfers for cycloproponations, for example, with iron-containing systems that catalyze that reaction, which is why we didn't talk about that. But and in some cases, they've seen that there's some carbene insertions, and maybe Jared Lewis has also seen this in some of the work that he's done of carbene insertions into the protein side chains. And that's a way you can modify proteins also. So we usually think about it as a way that they might be decomposing. In our case, we did do some mass spec, right? You can look for that by mass spec as to whether your protein has an added mass from the carbene, and we did not see that. I can't say we scoured and did absolutely the most careful experiments to check for that, but it looked like that's not happening in our case. But I hadn't thought about using that as an activation mode or some way that you could do something that you wouldn't naturally get from natural amino acids that you could use. So that's kind of a cool idea. Thanks. Yeah.
John, I have a more philosophical
Yeah.
question. There are so many molecules out there, and you can choose to put a bond somewhere on the molecule. I wonder, where do you get the inspiration to think about what kind of chemistry you develop or where you put a bond?
Right. So that's a good question, and we weren't so good at that in the beginning, maybe, of our career. Right. So I guess there's two questions in there. One is, how do you decide what reaction you want to catalyze? And two, if you have a reaction to catalyze, how do you know where it could be applied? Or because the first example of something you publish is usually not that great, and low turnover numbers, narrow scope. And so how do you decide where to try, where it's going to be applicable and how to develop that? And I don't know. I guess there's some experience. I mean, that's like the project selection is probably for all of us in the room is the major thing, right? So we try to develop reactions that fulfill two criteria. One, where there would be some fundamental, mechanistic question that's to be answered, or where it's going to be a reaction that could be generally applied, right? Something that wouldn't just be applied to one particular natural product or one particular medicinally active compound, but something that could be a general reaction, a general reaction of general use. That's one. And then how you decide to develop it is, again, talking to people, learning what their needs are, or taking, the Apple approach of telling people, developing something and telling them that they need it. A little bit like the CN coupling, where they say, why do I need that reaction? You know, because I can make things other ways. But then when you give it to them, they use it all the time. Yeah.
Thank you very much.
Yep.